So on that, I'd like to introduce Josh Walker, and he's going to go ahead and discuss two important components of this workflow. Speconsense, which is I'll let him get into the details, and then something that we haven't really used yet, but I think will play an important role as we move forward, a program called gapHACk. So without further ado, I'll leave it to you.
Thank you, Hart. I'm Josh. Let me see if I can get my screen shared here and get some slides up. Okay, do you all see something that looks like a slide? Yes.
Yes.
Okay. Um, so I'm going to try to run through slides in about 10 minutes or less, and then mostly show you how some of the tools work and answer questions. I'm sure you'll have plenty. Um, And how do I advance? There we go. Quick about me. I like mushrooms. Mushroom nerds are fun and weird, and I'm one of them. I've been on iNaturalist and sort of getting more into sort of deep identification for about the last decade and got into the MycoBlitz just recently in 2023. So that was my first foray into DNA barcoding. My background is software engineering, and luckily I was able to retire in 2020, so I have a lot of free time on my hands. Never done anything with bioinformatics or data science or formal mycology before that. But for the last couple years, I've been spending more or less half time working on tools, and I'll sort of tell you how I got into that. Specimux was the first program that I developed. So when I first got DNA results back from the MycoBlitz, somehow I ran across one of Stephen's videos on how he did sequence validation. And he was just, opening up the MycoMap site, and almost all of what he was doing was comparing the blast results to the photos on iNaturalist and throwing away, three or four that were off target and keeping the one that matched the specimen. And that um, seemed weird to me, seemed suboptimal. So I started up a Facebook chat with Steven, and he actually replied to me, which was nice, and sent me some details on how the data analysis worked. And importantly, he sent me a nice big file of FASTQ reads that I could play with. So I worked with Minibar was the program that everyone was using before Specimux. Worked with that and eventually wound up with some improvements that reduced the off targets pretty well. And I think Hart was excited about that. I think everybody was excited about that at the time. So I I'm not sure who all we have in the audience. Probably some people more into the bioinformatics side, some people more into the mycology side. I will try to explain the software geek terms that I go into. But the big thing that helped me at the beginning as I was trying to get improvements was just to be able to measure whether I was going in the right direction. So I took a data set from one run and curated all the correct sequences that should come out of that data set, and I tried to improve the precision and the recall against that data set. Um, I like small tools that do one thing well, and that's why we have a bunch of different names. Not everybody likes that. Some people like just one, dashboard that you can click on. Um, Reliability. So I'm really going for tools that work well, tell you what they're doing, explain the options that are available. And usually the algorithms are not very fancy. Usually I'm just putting pieces together that already exist. And what I've found is that It makes a big difference whether you use a fast building block or a slow building block. edlib, for instance, is a super fast alignment tool that can, align two sequences and calculate the identity. If you use that, you'll be able to do a lot more than if you use, say, BioPython's aligner component, which is great, is a lot more configurable, but is pretty slow. And if we use that for Specimux, the runs would take a day instead of 5 minutes. Um, and I'm not trying for a lot of fancy algorithms or data structures. For most of what we do, gapHACk is kind of the exception, but for most of what we do, you can just brute force it. You can just, compare everything against everything else, and there's enough compute power in the world to do that. Uh, I don't use a lot of compute though. I run on my MacBook. So that's only 8 cores, 16 gig of RAM. So my idea so far is if I can run it on my machine, on my laptop, then anybody should be able to get a decent experience out of it. So Speconsense is the specimen consensus generation tool. It's a replacement for what the pipeline used before NG species ID. And around the time that I was, exploring a replacement for NG species ID, a lot of folks in the sequence validation core were lovingly phasing haplotypes using Eugene and FASTQ files and just kind of pulling apart the reads, and that was interesting, but very manual and slow. Stephen Russell used AI to build sort of a proof of concept for haplotype phasing. And that was interesting. I didn't like all of the ways it worked. So I explored some alternatives and came up with this Markov clustering algorithm. That's another off-the-shelf piece. But it does clustering without a priori knowing how many clusters you expect. And that's different from a lot of the other hierarchical clustering algorithms. So I built a prototype. I'm sure I sent somebody the link, but it just kind of sat on the shelf for about 5 months until August of last year, when I think we had a sequence validation core meeting, and I mentioned it, and then Stephen picked it up and started testing with it, and had a ton of feedback for me. So I spent a lot of last fall, revising Speconsense, fixing it where it needed improvement. And the biggest improvement was phasing the variance in detail. So the Markov clustering algorithm happens to pull apart variants pretty well, but it doesn't get them all. And so what I added last year was a second pass that looks at the multi-sequence alignment, and looks for variant positions and pulls those apart. And I'll talk a little bit more about how that works. So I think the first runs on Micomap that use Speconsense were sometime in Q4 of last year, roughly, October, November. Steven started using it. So a real simple overview, and I'll try to show more in the demo of how Speconsense works, is there's two main phases. Phase one is clustering. And the idea there is to take the reads from the specimen and just find the ones that fit together, right? And it's sort of more of a decomposition. So we build a nearest neighbors graph, and I'll explain what that means. But basically, you calculate the identity between every pair of reads, and then you keep the five closest as edges in the graph, and that's the input to the Markov clustering algorithm. Uh, That algorithm gives you coarse clusters, and then there's a couple passes to uh, phase out the variant positions and then combine back anything that turns out to be identical or equivalent. So there's a little bit of iterative pulling them apart, putting them back together, and then generate the final consensus. So the idea with phase one is to pull apart all the variants that have good support. Phase two is summarization. And when we first started looking at how many variants come out of a typical specimen, I think Hart and Stephen and maybe a couple other people were looking at it, and we just said this is too much for everyone to look at and deal with. So a lot of the options in Speconsense actually are aimed at trying to either choose which variants to keep or conflate them back together so that, if they only differ by one polymorphic position, just merge those together with an ambiguity code and keep one sequence instead of two because they're close enough and you don't want validators to have to review every single sequence individually. That's one area that I think will probably evolve over time. There's a little bit of codependence with what MycoMap supports in terms of collapsing and visualizing multiple variants. As that gets better, Speconsense can probably produce more variants, and we can have those preserved in the raw data, but only look at the summaries on the front end. I think I'll run through the rest of the slides, and then I'll be interested in what questions you all have, and we'll do some demos. So I got a couple slides on gapHACk. gapHACk is much less mature than Speconsense. I started working on it when nobody was looking at Speconsense, when I had a little bit of extra free time. And the goal was to rather than working with the individual reads for a specimen, work with the consensus sequences. So, imagine all your blast results for a particular sequence. Work with those sequences from multiple specimens and try to find the natural boundaries between them. You can think of it as sort of trying to automate the temp code delineation process, right? These things go together, these things don't fit. So where is that boundary? And there's published literature on the barcode gap. And the idea here is if you see this little histogram, the blue are intra-cluster distances and the red are inter-cluster distances. So the idea is that if you've got, a well-defined cluster, there should actually be some clear separation between the distances within the cluster and the distances outside the cluster. And that way you can say, well, if it's more than, 0.7% difference in identity, then it's probably a different species or a different OTU at least. So that's what gapHACk tries to do. Um, it's um, it's a fairly simple algorithm, but there are a few different parameters in it, and it's pretty sensitive to exactly how you set those parameters. And I think that'll be clearer when we actually look at a few examples. So I think it's a promising tool. It's useful as long as you're going to review the output. It's not to the point now where you can just, trust it without looking at what it produces. So that's sort of where we are. Specimux is pretty much bulletproof now. I think, we'll get to a 1.0 release pretty soon. Speconsense is very reliable, but there are a few things that need tweaking, mainly around which variants we keep. And gapHACK is still experimental. You're welcome to play with any of these, but I will respond and try to fix bugs on the first two. And usually I'm pretty quick on getting bugs fixed. With gapHACK, I'm just probably going to say, well, I don't know. That's interesting. Let's play with it. So in particular, the things that I'm focused on right now with Speconsense, the variant threshold. So what's a significant variant versus a potential artifact? I think I have a good direction on that, and we'll probably get that out in the next, month or so. I may not hit micromap sequences for a little while, but that would be pruning the really low support variants and giving us all confidence that what remains is not a bioinformatic artifact. The other thing that I'm really excited about with Spec and Sense is looking um, more closely at what we do with homopolymer normalization. So when you have a run of the same base, um, nanopore is known to have a higher error rate in long runs of the same base. We use adjusted identity in a lot of these tools, which treats any difference in homopolymer length as equivalent. And what my initial analysis is showing is that we can actually trust nanopore's homopolymers up to about length 4. 4 or 5 is where it really starts degrading in a nonlinear way. And so what that means is we'll probably, at least as an option, be able to phase variants that are off by just a homopolymer difference. And that can be useful because some of those are pretty clearly biological differences and not just nanopore artifacts. With gapHACK, the ultimate goal is to take all of the, 100 and however many 100,000 sequences we've got from North America and recluster them algorithmically and see how closely that matches the temp code boundaries. Um, I think in general, the folks that have been doing validation, creating temp codes do an excellent job. But the data on Micromap gets pretty noisy where, a button doesn't get clicked right and a contaminant winds up with the primaries label. And some of that starts to really pop when you run a tool like gapHACK across it. And then not so much related to those two, but Specimux Suite is a higher level workflow automation that lets you just run one command and it runs soup to nuts and shows you in sort of a live dashboard what sequences you're producing and what the tentative IDs for them are. So, there's another photo for your interest. Let me see. Let me get out of my slides. And have there been any questions in the chat yet? Or anybody want to speak up and ask a question before I show some examples? Okay. Then I will just show you a few things with these tools. Um, Can you see my terminal okay? Is it too tiny?
It's okay for me, but it might be a little small for
Yeah.
some people.
Let me make the font a little bigger. Um, that's bigger. Let me know if it turns out to be a problem for anyone. I've got a few different things here. I thought first I'd show just Specimux and Speconsense so that you get a flavor for how these sequences are produced. So that's in this folder called demux. And let me show you the source data. Um, so there's 3 files that go into Specimux. There's a primers file, which is usually the same. And that's just, the name of your primer, the actual sequence for it, and then whether it's in the forward or reverse position. And Specimux supports primer pools where you can have, multiple forward and reverse primers together if you want. The other input is a list of specimens. So you got a name for your specimen, what pool it's in, the index. So this is the unique tag that goes on each end of the read. Your forward primer, the index on the reverse end, and your reverse primer. And then you have a bunch of raw reads, and I don't run the nanopore tools myself. I just get data from Stephen or Hart and play with that. So I start with usually a FASTQ file or a BAM file, but those just look like this. So probably everybody's fairly familiar with a FASTA file. A FASTQ file has this extra thing that looks like garbage, but is just the quality score. And actually, all of the tools that I've built so far mostly ignore the quality score. There's a little bit of sampling that uses the quality score, but it's really not a big input. So we could do most of what we do with just the FASTA file. Um, so that's sort of the data. And then what I'll show you is just a little bit of how Specimux runs, so you get a flavor for it. So Specimux is available as a pip package, so you can just do pip install Specimux if you don't have it. There are a couple dependencies you need, and those are documented on the GitHub page for it. So there's installation instructions on GitHub if you actually want to run Specimux yourself. So you run Specimux with the primers, the specimens index, and the raw calls. I'm just gonna let it do the first, say 10 here, and I'm gonna do color mode so that we can see, and we're not gonna trim anything. So let's go back to where we started. So it calculates some distances, but basically, if you can see the color coding, this is what it does with the reads. So it finds the primer on each end, which is in green here, and then it looks immediately adjacent to the primer for the index, which is in blue, and then it matches that to a specimen. So this forward and reverse primer happened to be from 1.85. Um, and then, this next one was from 10.82. And some of them won't match on both ends. So if you see this read here, it looks like it had a reverse index, which is under where I see you all, under my Zoom window, but it didn't find a forward index here. This wasn't close enough to match a forward index, so it just stuck it off in a partials file. So that's sort of the job that Specimux is doing. The default trimming keeps everything with the primers and everything in between the primers, but it removes the indexes. And so that, if you go nowadays and you download an assembly file from Micomap, you'll get reads that look like this. They still have the primers attached on either end, but they don't have the indexes. So we don't run it like this normally. We run it so that it dumps everything to a separate file. And we've got about 1 million reads here. That's going to take roughly 5 minutes. We don't have to wait for that to finish because, like any good cooking show, I prepared one ahead of time. Um, so when Specimux finishes, it gives you a folder that looks like this. So it gives you all your full matches, and we named our pool ITS. So that's where it dumped all the files. And you get one file for each specimen that was in your input. So what do these look like? So these are much cleaner. So let me look at it this way. Let's look at just the first one. Um, and let's look at just 100 reads out of that. So I don't like opening up Eugene. It's way too slow for me. Um, Let me get this so that it's a little bit more legible. So I have a little command line tool that I use um, that just shows the variant positions in a file. So here I filtered down to everything that's within 5%, at least 5% different. And you can see that we're just omitting the bases that are identical across all the reads. And we highlight the variant positions here. So this gives you an idea for what's in the file, and you can still see there's still probably some nanopore errors that might be showing up here. It's hard to tell if all of these are biological variants or if some of them are just random errors. I'll show you one more way of looking at the data. So the same first sample, 01.01. We'll just look at 100 reads again. So this takes just a second to load. So this is just an adjacency matrix, right? So every cell here is an identity value used, comparing two sequences just like BLAST would do, and we can drill down on one of these and see an alignment that looks like BLAST results, right? And this is just the grid of the 100 reads that came out of that FASTQ file. And you can see a lot of them align pretty well with each other, but there's some down here where, this is probably not the same thing. It's only 80% identical. There's all kinds of mismatches all over the place. So that's probably not our target for the specimen. So what that's sort of the way Speconsense views the world. And if I run Speconsense, and let me make a little work area here. Um, so Speconsense takes just a FASTQ file as input. So if we give it that 0101, and the defaults should work fine. It builds that adjacency matrix at the beginning, and then it runs the MCL graph algorithm. It came out with 89 coarse clusters from that first pass, and it checks to see if any of those are the same or equivalent if you ignore homopolymer differences. So 19 of them got merged together, and then it looked for variant positions, and it looks like it didn't phase any variants there. So it stayed with 70 clusters. It filtered the ones that were tiny, and there are 9 clusters remaining. And so what it did is it created an output file here um, that has just the consensus sequences along with some metadata. So in the FASTA header line, you have the size, how many reads it actually found in the cluster, the reads in consensus. That's how many reads it used to make this consensus. It subsamples by default to 100. Um, these other fields most people probably don't see most of the time. RID is read identity. This is calculating every read against the consensus, what's the net identity? So there's a 0.3% deviation on average between the read and the consensus, which is pretty good, right? And those deviations average out and get corrected in the consensus anyway. This is just looking to see if you have a cluster that's not very cohesive. And then the RID min is for any single position, what's the maximum variation or what's the worst identity for that position. So for this particular consensus, there's some position that's 1.7% different. If we um, if we look at these now in a grid, um, you can see this is just the consensus sequences that were produced, and these are essentially each of the variants compared against every other variant. So it's a pretty tight set. There's not a lot of variation here. Some options on Speconsense. So most of the tools have a dash dash help, which gives you all the command line options and some short text if you're not, if you don't want to go over to the GitHub page and read the README, which explains these in more detail. If you just want to get an idea of what you might play with, dash dash help is a good way to start. And if we do this again, so this is the way I ran Speconsense before. We can say uh, sample more reads. So the default is 1,000. We can pre-sample more. We can generate each consensus with fewer. Once we make the cluster, it only takes probably 10 reads or less to get a good consensus if they belong together. If they don't belong together, you get a garbage consensus. This is showing something that we mostly don't use, which is scalability mode. So Speconsense now, you can actually run it against up to 1 million reads in one specimen, and it will work and produce clusters. And I can talk about how that works later. So um, that's a little tiny flavor of Speconsense. Um, what I did that sparked this talk was, let me see if I have it here. Bear with me for 1 second. Um, so I sent this across to the Slack chat for the sequence validators, and this Claude writes beautiful LaTeX. This looks a lot fancier than it actually is. This was a quick analysis I did looking at Amanita lavandula and reprocessing old specimens. So unfortunately, um, it's hard to replicate this today, But what I did is I have a little scraping tool that looks at iNaturalist, finds all the observations of a given taxon that have barcodes, and then looks for micromap links there and tries to download all the FASTQ files. Why I say you can't replicate this anymore is I guess Steven had a scraper running against the site yesterday, and so he implemented some anti-bot tooling. And now if you try to go get the trace files, you have to prove that you're a human. So my scraper doesn't work anymore. Um, but if everybody has seen Micromap before, this is where you get raw reads that made a sequence. All right. So if you get to this, I don't know if this is called the accession page, but you click the assembly files, and that's going to download raw reads for you. Um, and what I did here was a couple weeks ago, I scraped all the Amanita lavandula sequences I could find, got their raw reads, and then reprocessed them. And that's one of the reasons you might run Speconsense yourself if you're not, if you're not doing nanopore sequencing in your own lab like Hart does or Stephen does, or I think I may have 4 or 5 users worldwide of these tools. Um, but if you're not doing that, one thing you might want to do is download older data from Micromap and reprocess it. And I'll show you one important bit with that, um, which is when you're reprocessing, let's say, one of those files. Let's actually find the biggest one. Okay, so this looks like the one that has the most reads. So if you're going to reprocess, you need to reorient the reads normally. So I forget the name of that option. Let's check orient mode. So the reason for this is I can't type and talk at the same time. Is the old data before Specimux, so roughly probably before 2024, used Minibar. And Minibar was another demuxing tool. And one of the key differences between Minibar and Specimux is that Minibar left the reads in their original orientation. And so when you're sequencing with Nanopore, sometimes the molecule will go through from the 5 to 3 direction, and sometimes it'll go through from the 3 to 5 direction. And with the old data before about 2024, they're all mixed up together. So with the newer data, Specimux normalizes the reads, so they're all in the same orientation after Specimux, and all of the new files on Micromap should be in the same orientation. But if you want to reprocess an old file, you have to tell Speconsense to try to reorient them. And here I'm telling it just to throw away the ones that don't work. So that's what filter failed means. So this is just running Speconsense like we saw earlier. And we got four clusters out of that. Um, and here they are. And you can see the size drops off pretty quickly. We had 400 reads in the first cluster, and then just 26 and 11. And that one, the 11 and the 5 are probably not significant. Um, so what can you do with this? Well, The interesting thing I think about being able to phase out the variants is that when you have this intragenomic variation, it's sort of a toss of the dice what sequence you get with the older tools. Um, Usually it looks like you get something that's pretty close to the most abundant variant, but it can have sort of chimeric indels or substitutions from other variants that are in the specimen. So if you phase out the variants, then you've got just, something clean that's presumed to be biologically real. And for the groups that have a lot of, large variants like Amanita, uh, it's interesting to look at the co-occurrence, right? So do you
Yep.
get, sorry, was there a question there? Okay. It's interesting to look at, which variants occur together in the same specimens and which ones are distinct. And so that's what that little write-up on the Amanita lavandula variants was about. Let me shift to gapHACk real quickly. So I'll use some different data with gapHACk. What I have here are just a handful of FASTA files that have all of the mycomap observations that are labeled with one of these taxa. So I think Colibia nuda was the one, the bluet was the one that I thought showed something cool when I first developed gapHACk. So. So gapHACk works up to about 200 sequences. It depends sort of on how much variation there is. If there's more variation, it's more likely to be slow. But let's run it with that set of sequences. And it does a similar sort of thing. It uses the adjacency matrix, and it has two phases. It has a fast phase and a gap optimized phase. So if we ran all the Calibian nuda sequences I could find, and then it produced three files here. A cluster one file, a cluster two file, and a singletons file. And if we, I think I need to get the right tools in my path. 1 second. Okay, so if we look at those and look just for the major differences, um, there's actually quite a few. Um, And some of these are just alignment artifacts because some of these sequences are full ITS and some of them are ITS2. And so they don't always line up really well when you're doing that. Um, if I look at, let's look at the distances instead. Um, So this is where I think you can see clustering sort of intuitively. This is again just the distance matrix, but it's sorted, and you can see there's this group in the upper left that are all pretty similar to each other, and then there's a group in the lower right that are similar to each other and they're fairly different, right? So they're about 2% apart. And the interesting thing with these is if we look at look at the two clusters. So here's a map of the first cluster. And if we open the second cluster, they're geographically distinct, right? So we have a West Coast bluet and an East Coast or Eastern North America bluet, and they have a little bit, about 2% of ITS variation between them. Interestingly, these don't have separate temp codes right now. We're calling them all Calypia nuda with the full binomial. There's also a couple singletons that did not cluster with either of those. So singletons is what it calls the sequences that, don't have any peers. And some of those were probably on Mushroom Observer rather than iNaturalist. This is the one on iNat, and it's actually the Cypress bluet, right? So there's one Cypress bluet that somehow wound up as Calypia nuda in my data set. Okay. So that's that's what gapHACk does. As I said, it's a little bit sensitive to a few things, and let me show you the major ones. So when we ran gapHACK um, on Calypia nuda, there are two parameters. Uh, One is called min split, and this is basically the smallest difference that it will separate into two clusters, and you tell it that. So here, the default is 0.5% difference in identity, right? So if two sequences are at least that similar, then they always go into the same cluster. gapHACK will never separate them. And you can change that. You can change it to 0.2% if you want. And the other big parameter is max lump. So this is the largest difference that it will ever put together in the same cluster. So the default is 2%. Anything that is more than 2% different would necessarily be separated into two different clusters. And you can change those. Hopefully, they don't change the outcome, right? That would be nice. That would mean the algorithm is stable and independent of the specific parameters you give it. In this case, I think we'll get the same result, but in other cases, it does matter. And you can see it's a good bit slower this time, and that's mostly because min split is set smaller, which makes it consider more ways to cluster. So we got a slight difference here. We got three clusters instead of two, and and we got more outlier singletons. That's probably from the split difference rather than the lump difference. Okay, I'm gonna stop talking. I didn't really get to show you a whole lot so far, but I'm interested in what questions people have.
Thank you. Looks like there's a question in the chat from Keith. It says, would eliminating low Phred score bases in pre-processing help reduce the amount of sequencer errors in the polymorphic sites? And this was from about a half hour ago.
Okay. Um, so I think the answer is maybe, right? So I don't completely trust the sequencer to know when it's right and wrong. And that's why I kind of ignore Phred scores entirely. The very first thing I did with Specimux was try to use the quality scores to choose which reads to bucket. And I didn't really know much about what I was doing at that time, but it did not make a positive impact at that time. Um, Nanopore error now is really quite low, right? I think the published error rates with the current chemistry and the current base calling is something like 1.5%. And so with 1.5%, if it's randomly distributed error, you only need a few reads to average out those errors. You know, like I said, five is enough. 10 is more than enough. Um, and at that read count, you can start to phase out the biological variants as well. So it's sort of our where we started, the our bias was to keep more data and not throw away data that may be significant, and that's why we didn't really throw away reads with low Fred scores. The sampling does sort by the average Fred score and keeps the ones with the highest average Fred score. So if you have 2,000 reads for your specimen and you sample 1,000, you'll get the 1,000 best reads, supposedly.
Yeah, and I'll add to that a little bit. You know, even within the confines of the current nanopore chemistry, the sequences we generated a long time ago did have lower overall Fred scores, and that's evident in doing alignments. You can see that they're much noisier. Although I think that that comes down to the base calling algorithm. So when we say raw data for the FASTQ files, that's a little bit misleading. Nanopore outputs this trace file that's uh, electrical signals, and that gets interpreted into what we then use as the FASTQ file for the demultiplexing. So it is kind of a black box as far as being able to look in and see what, you know, how they're calculating the Fred scores. So I think that's also part of the reason why it's hard to even um, truth whether they're useful or not because you don't know how they're assigned, if that makes sense.
Yeah. More questions? Other scenarios people want to look at?
Maybe
Um,
on the same topic, is this also how the reads in the cluster are chosen based on the ranking of Fred scores? If you have 500 reads in a cluster, or 500 reads in the total size of the cluster, and then only 100 reads end up in the final cluster, is this also how they're prioritized?
Um, I think so. Um, let's ask Claude. That seems like what I would have done, um, but um, but I don't remember.
Mhm.
I also don't think it makes a difference because once you get the cluster, you say you have 500 reads, um, there's pretty high confidence that those all belong together, and you're sampling just in order to average out the errors, right?
Mhm.
And so, really, the abundance matters more than the quality. But yeah, it looks like we do sample. Anytime we sample, we sort by the mean Fred score.
Mhm. Cool, thanks.
Yeah.
I had a question on the tip of my tongue, but if anyone else has more pressing questions, I'd let them go ahead of me. Um,
Go ahead.
so I've noticed that the output that then goes into Micomap these days, I want to understand it a little bit better. So sometimes I'll see IUPAC ambiguity codes called, sometimes I don't. And in general, the FASTQ files only have 100 reads. Um, and I don't exactly know what is creating that particular package that's currently being uploaded to Micomap. So um, when does it decide to put an IUPAC ambiguity code? When does it decide to split it into a unique grouping, and sometimes I see FASTQ files with 200 reads. So I just could you explain some of that a little bit?
Yeah, good question. Um, so there's two phases to Speconsense, and I really only showed you one. So when you run Speconsense itself, you get you get consensus sequences for every variant or everything it thinks is a variant in the specimen. And um, in general, those do not have a lot of ambiguities. They have some. And so the way that's controlled is um, there are these limits on variant phasing and ambiguity calling. And so with variant phasing, there's a minimum frequency which defaults to 10%. And I think Stephen is using the default. And there's a minimum count, right? Which is five. And these were my initial way to figure out if a variant is significant or not, right? Or just to put an interpretable threshold so that we can say, well, if it's 10%, anybody would reasonably say that that's a real variant. Um, what I'm working on right now is to put a statistical framework in place so that we can actually look at the size of the input pool and the size of the variant and throw away or not phase things that don't look significant. There are separate thresholds for ambiguity calling. They're the similar concepts. So a frequency and a count, right? The count for ambiguities is smaller. So in the first phase, where when it's trying to find the variance, it will split the clusters recursively. So it finds what looks like a variant position. It splits that into two clusters, and then it looks at each of those clusters to see if there are more variant positions at the 10% level just within that new cluster, right? That smaller split cluster. And it recursively does that all the way down until it hits the limits. Um, if it hits a limit and there's still a minor allele that exceeds the ambiguity frequencies, then that first step we'll call an ambiguity code, right? And that if you see ambiguities in this cluster's output, those are more or less true ambiguities, right? They mean that we didn't have enough read support to phase out the variants that probably exist. Um, but probably they're low abundance variants. The second phase is a tool called Speconsense Summarize, and the job of Summarize is to conflate variants back together, right? And this is where a lot of the bugs in the first few runs were. And the the goal with conflating the variants back together is just to give people less to look at, right? And so a lot of the ambiguity codes in the early runs that shouldn't have been called but were was from conflating a really high abundance cluster with a low abundance cluster. So, you'd have, a cluster with 500 reads and a cluster with five, and they would get conflated together as peers, and an ambiguity code would get called, right? Um, let's look
Can I
at,
ask a
yeah,
question about that?
yeah,
Um, how was that? I know that's fixed because
yeah.
I encountered that a lot in the beginning and now I don't. But what like sort of weighting parameters were set to like before an ambiguity code would be introduced?
So with the summarize step, um, the sequences are always treated as peers. So they're not always merged now, right? There are some limits on what will merge. But this is uh, I will call it an abuse of ambiguity codes, right? What we're doing with summarize, because there's no ambiguity, right? We know exactly what the variants are going into summarize. And we're using an ambiguity code to say we saw both, right?
Yeah.
And so that didn't really have, doesn't have, still doesn't have limits. You know, if it's 100 plus 20, here's one example, it's going to use the ambiguity code for whatever that position was, whatever the two bases are. Um, there are now limits on what will merge, and the main one is a ratio, if I can find it here. Um, this merge min size ratio. So anything that's more than a factor of 10 different in size will not merge together anymore. And that was the main workaround to get rid of that low support bug. So that's set to 10% to mirror the ambiguity calling in the first phase. The idea is, you know, if you open up the FASTQ file for those two merge sequences, you should see at least 10% variation. And so it should sort of pop to your eye now.
Yeah. Okay. Uh,
Um, yeah, go ahead.
sorry. Am I right that there's also a limit that a maximum of 5 ambiguity codes could be used in a single file? Is that still in place?
There's a limit to how many variant positions. There are many limits to when sequences will be conflated, right? And they're aimed at um, not destroying too much information, right? So so what you're thinking of, there's merge position count. The default's two. I honestly don't know what Steven uses. I think it's probably a lot higher than two. Um, but this,
Yeah, we see more than two sometimes.
yeah, this limits how many positions can be different before sequences will be conflated.
Okay.
And the reason for that is if you have, you know, three variant positions or three ambiguity codes, there are eight possible variants that come out of that, and you probably only saw, you know, two or three. So you're saying, you know, with three ambiguities, you're saying eight, eight variants were observed, but really that's not true, right? So that's where we're abusing ambiguity codes a bit. The defaults also will not merge anything that differs by an indel because you can't represent that in ambiguity codes. I don't know what Stephen and Hart are using, if they turn this on or not. Um, so this is where, you know, I think there's too many options for choosing what to merge and what not to merge. And that's sort of because we're still in early days and we need to. It's also because not everybody agrees on what we're trying to do, right? Some people want to knock it all down to basically one consensus sequence that represents everything. Some people want to see a dozen different variants, and so the tool supports all of those use cases, but it probably needs to have more opinions about what is the right way to do things.
I think, um, you know, the strategy that I taught people with sort of relies on the idea that within NCBI GenBank, there's already some fake sequences in the sense that whenever someone's interpreting a Sanger chromatogram, you're automatically conflating sequence variants together because there isn't a reliable way of separating them. So that combined with the adjusted identity metric in Stevens blast search means that if you do merge two different variants, even if you're merging indel variation as well, and you do a mycoblast search, um, because often these indels are not considered significant differences, it'll give you 100% adjusted match to representatives of either of the variant groups across MycoMap. So it's kind of a way of seeing everything that comes into a pool of variants by only looking at a single sequence and only comparing with a single sequence. So what I have a gapHACk do is essentially that it creates a merged consensus. Um, but ideally, we want to move to a future, at least this is my opinion now. Ideally, we want to move to a future where we're looking at the accurate representations of the data. However, I wonder if there is a way, because I haven't, for clients that use my service, I haven't switched over to using Speconsense yet because I can't necessarily control the output to have it arrive as this merged consensus, which includes indel variation and separated sequences. So I think that's the one thing that's holding me back from fully switching over. And the other thing is, when there's FASTQ files that are pared down to just the individual variants, I wonder if there's a way to also include a more accurate sample of the original pool that people can use if they want to open it up in a manual alignment program and try to see if the results match their expectations given like a manual alignment.
Yeah, I think that's MycoMap is good at giving access to sort of the more processed data, but it doesn't really give access to the raw data that came out of demultiplexing before it went into clustering. And I think that's an enhancement I would really like to see to make it easy for other folks to go back and reprocess specimens with newer bioinformatics tools. Um, you know, for for you just working, on your computer, you can distribute the output of Specimux before it goes into Speconsense if you want.
That is true, yeah. That does Specimux have like a length filter built into it? I noticed one of the things I haven't looked at one of these demux files in an alignment in a long time, but some of the things I would see from minibar would be like, for example, just oligomers that are like primers stuck together. That would only be like 100 base pairs, or weird chimeric sequences that included, a chunk of completely off-target data in the middle of the sequence. And that would end up getting fixed by adjusting the parameters in the consensus building. But I wonder if is there anything that's filtering those out in Specimux?
You can. So there's a min length and a max length option on Specimux that will filter the reads before it even demuxes. Um, I I think I prefer to have those just fall out during spec and sense clustering. Like the old analysis protocol had I forget what tool it was using, but filtered for length, and you know there were multiple reasons why we weren't getting chanterelles. One is, of course, the primers don't amplify them as well, but the other was just that we were filtering out everything above like 1,000 base pairs, and most Chanterelle ITS is longer than that, right? So it's certainly like if you put a max length, but it's up to you. Um,
That's um no, that's a good point. Uh what was it? I was gonna ask you something else, but I got distracted. Oh yeah, so the other question I had was so I've been filtering the top 500 reads by quality score before going into Specimux, before going into Speconsense or any sort of clustering process. Do you recommend now just letting it stay at the 1,000 read um, default?
um, it's it mainly depends on how much you care about minor variants and contaminants. I think if you're just doing like normal sequencing of relatively fresh specimens, then 500 should be plenty to get you, your primary target with maybe a couple variants and any major contaminant you've got. Um, and it does get slower when you use more reads, right? 1,000 is the default, and it's not too slow for me. Like my Speconsense runs, if there were 1 million reads going into Specimux, it probably takes me an hour to run Speconsense across all those, which is fairly tolerable on my machine. But if you use fewer reads, it'll go faster. One of the things I think I will do with this new workflow tool with Specimux Suite is do sort of adaptable sampling based on whether the primary target has been recovered or not. Right. So far, all the tools are reference free. Like we don't make any assumptions about what sequence we're supposed to get. But I think there would be some value in saying if we've got, good hits to genus level, we don't need to use, all 2,000 reads. But if we don't, then maybe we dig deeper. Maybe we even dig into the partial match files and try to augment from there. And so that might help normalize, some of the amplification difference.
Yeah, that's something that we could probably spend a lot of time on would be some of the other outputs of Specimux, the partials and unknowns, and how to sort of mine. I think you have a tool called Speciminer. I don't know if that's still applicable, but how to sort of, because my demux rate in my last run was like 48%. Um, you know, there's probably a good number of the sequences that didn't demux that are just garbage, chimeric, random noise. But I bet there's quite a few in there that, for whatever reason, are just missing too much of the index to properly demux, but could still have valuable data. Uh, anyway, I unfortunately I've got to run to a board meeting, and I have to get in my car and drive there. So I will have to call the meeting, but I think this was really informative, and I'm gonna post it on the YouTube channel that I posted in the chat. At least for now, we're trying to get a DNAMA official YouTube channel, but for now, I'll put it on that account, and I'm recording it locally on the machine, so it'll take some time to process. And I'll probably trim the first, 10 minutes of like sitting around or whatever. But this was extremely helpful. And, one thing like to maybe set up in the future, especially for folks like the Quebec crew, who I'm not sure if anyone's on from there currently, would be a workshop um, where it would be more focused on actually using our data and going through and learning how to do different um, analyses and things like that. I'm really excited to see gapHACk when it's fully bloomed, because I think that'll really help us start to see the forest through the trees when it comes to distributions of taxa and distributions of variants within taxa, and really excited to see the full pipeline addition that actually does the uh, um, you know uh, taxonomic identification as well. I think I saw a demo of that. So I think there's plenty of opportunities for us to do some sort of future uh, workshops or meets. But did anyone else have any quick questions before I jump out? And I've
Are,
given them.
are
Go
are
ahead.
we already seeing sequences like variants being separated within MycoMap associated with certain records? Or is that like is it that when these variants have been clustered, are they being given individual accession record numbers within MycoMap?
Yeah, I think most of the recent runs have variant phasing enabled, at least the ones that Steven has done. And you'll see sequences that have a suffix like .v1, .v2, which are the variants that Speconsense pulled out.
Awesome. I was like seeing that and retrospect had to change some things because I was like, oh, there's other variants within this uh, group. Yeah. So thanks.
Yep.
And thank you so much. That was very interesting.
Yeah, so
Hey,
I'm
you're
I'm going
welcome.
to jump off,
Yep.
but I've left the host to Josh if other folks have any more questions. So
Thanks, Hart, for organizing this.
thanks for jumping on.
Yep.